I'm now going to give you an overview on what happens during transcription and translation. Do not confuse transcription and translation with DNA replication, because replication is the copying of DNA. And transcription and translation are both necessary to make proteins, because the real job of the DNA is to code for proteins, to tell the cell what proteins to make. And what I have over here is I have a cell over here, the green circle over here, and here that's our nucleus. This here is the cytoplasm. I don't know if you want to label this. This whole thing is a eukaryotic cell. And inside the nucleus, of course, what we find is we find our original DNA. And I'm going to just draw this schematically like this. That's supposed to be our original DNA, and it's of course much longer. And it's DNA. It always makes these spikes, and I don't really know why. What happens in the nucleus is that a part of this DNA is copied. And let's say that we're copying from here to, okay, from here to over here. And this is called a gene. And this gene codes for one protein, and this gene is now copied, and so I'm going to redraw this again, and this is called now mRNA. I'm making a small m, and this M stands for messenger, and RNA you have to write with capital letters. And this process, when the cell or enzymes copy a part of the DNA to make mRNA, this process is called transcription. And in eukaryotes, this transcription, of course, occurs inside the nucleus. So that's, and I'm only, as you can see over here in this mRNA, I'm not drawing a double helix, because it is not a double helix. It's single-stranded. And if you want to know the differences between DNA and RNA, there is a separate video to explain these differences. Now, of course, it's not only copied once, but it's copied many times. So you have many copies of mRNA. So I might want to write down here many copies. Many copies. Okay, so one DNA or molecule over here, one gene, that's probably better. One gene can be copied many times and have many of these same mRNA molecules. And what they do now is the following: they leave the nucleus, because inside the nucleus, not drawn over here, but they're little holes. And we don't say holes; we say that these are nuclear pores. Nuclear pores, actually, nuclear pores, and because what we have over here is this is called the two nuclear membranes. Two nuclear membranes. Okay, and the mRNA, what it will do is actually it will go out these nuclear pores into the cytoplasm. Okay, and in the cytoplasm, what I now have is I have the so-called the ribosomes. And the problem that I have right now is that these ribosomes are really tiny. So actually, if I draw a dot like this, then I don't know, probably a ribosome is even smaller than that. Yeah? So I cannot draw it because it's too small. So I have to make it too big. So if I draw it right now, just understand that I'm drawing it way too big, okay? Because I just want to get the idea across. So what I have over here is this mRNA molecule right now. That's the one which just left. I just label this mRNA, the messenger RNA. And then I basically have the ribosome sitting on here. And the ribosome has a small and a large subunit. It's not so important for right now. That's the small subunit, and there's a huge, large subunit of the ribosome. Okay, so I should label this as ribosome. And this ribosome, as you probably already remember, is responsible for making proteins. And how does it do that? The ribosome now travels along this mRNA, and it will basically read off the bases, because they're the bases, the mRNA bases on here. It will read them off, and it will insert a correct amino acid, because remember, a polypeptide or a protein is made of amino acids. So what I have is over here, these little circles over here. So that's a little bit of simplified drawing, okay? These are all the amino acids, AA, but they're not alone. They're linked to tRNA. I do not want to confuse you right now, but it's actually part of a different video. So it's not just the amino acid, but they're linked to a tRNA. We don't worry about this. In any case, these amino acids basically now go into the ribosome, and out of the ribosome, basically the ribosome connects it, and what comes out here, that is the polypeptide chain. That's the protein. I don't know what else does these weird spikes. Okay, that's the polypeptide chain. So in other words, the ribosome uses the information on the mRNA to attach the correct amino acids together over here. Some people want to know now, how does this actually work? Is the mRNA somehow converted or changed to a polypeptide? No. It's not converted or changed. It's just read off. And based on the instructions on the mRNA, it makes a new polypeptide. So this mRNA can, of course, be used to make many polypeptides. So that is basically the idea. And this process here, which I have drawn way too large and is not detailed enough yet, but this process is called translation. Translation, because essentially we're translating, or the cell is translating from base sequence, that's the base sequence or the nucleotide sequence, to an amino acid sequence, because these are now the connected amino acids. And that's basically like you're translating from one language to another language. So, and how this works is a totally separate video. I just wanted to give you now an overview in a eukaryote where transcription and translation happen. Now, in prokaryotes, which do not have a nucleus, the situation is similar but not quite the same. If this is a prokaryote, you see that there is no nucleus, and essentially I'm going to draw now the DNA over here, and the DNA is a long. That's that's the worst DNA molecule I've ever drawn. That's supposed to be the double helix, okay? I don't know, this is so bad that I actually would like to erase it almost. Okay, so that's the double helix, that's the DNA, and basically we have these genes on here. Okay, that's a gene. And what happens is the following, that this DNA is now copied, and the mRNA is made. It's still basically copied over here. It's made, but as soon as it's already made, the ribosomes start to attach to it. And these ribosomes read off the mRNA. It's way too small. I'm drawing this way too small. And then out here comes the polypeptide. Yeah. So we have, you know what? I'm just going to draw this again because this is not okay. Okay, let's make this. So that's supposed to be the DNA. It's supposed to be the double helix of the DNA. You get the idea, okay? This is a gene, and what happens is the following: as it is copied, and as the DNA is copied, the mRNA is made. So that's my mRNA. Yeah, so this has already been copied, and over here you have the so-called the RNA polymerase, which copies the DNA to make mRNA, which transcribes, not copies the DNA, well, transcribes the DNA. And over here on this mRNA, I have my ribosomes. And these ribosomes already, even before the DNA has, even before the mRNA has been finished making, okay? It's not finished yet, it's still in the process, but already these ribosomes start to attach to the mRNA, and they start to make a polypeptide chain, okay? And this is basically when we see something like this with all of these ribosomes attached to the mRNA, we call this a polysome. Okay, it's simply the name, but the important difference is that in a prokaryote, transcription is up here. Yeah, that's transcription, and this over here is translation. Transcription and translation occur both in the cytoplasm. So there is no separation between transcription and translation, because in eukaryotes you have the separation, because transcription occurs inside the nucleus and translation inside the cytoplasm. But over here, because we don't have a nucleus, it's kind of all in the same space. Okay, so that is essentially that's basically the overview of transcription and translation. And in different videos, I'm going to be talking in more detail about these two events.